Hello, I'm Ann Montgomery, Editor-in-Chief of Bioprocess International magazine. I'm here speaking with Dr. Kavita Iyer, who is the Associate Director of Global Regulatory Affairs and Clinical Safety for Vaccine, CMC, at Merck. So welcome and thank you for agreeing to speak with me.
Thank you, and I really appreciate the excellent opportunity to share the experiences that we have through BPOG closed systems.
Thank you. Let me ask you first to describe what BPOG is and what led you to participate in it, and how that participation helps you as a regulatory affairs professional.
Sure. So to start with, what BPOG is also known as the Biopharm Operations Group. So BPOG, it is an excellent forum and a powerful consortium of leading biopharmaceutical industries that actually connect, collaborate, and accelerate biopharmaceutical manufacturing processes with specific emphasis on process and product quality. So it's a nice platform wherein you can share ideas, benchmark, and also collectively develop industry best practices, which is extremely important to proactively and potentially influence the regulatory expectations and shape health agency guidances. So as a regulatory professional, I've been very intimately involved into the Biopharm Operations Group for the past 4 years, and primarily in 3 work streams, which is the closed systems, the post-approval strategies, and the regulatory interaction. So my experience has been, it's been extremely rewarding and satisfying, wherein I've had the opportunity to work with several industry experts and actually lead efforts in collaboration with them to proactively influence and shape health agency guidances, and also continue to strengthen and enhance the health agency industry relationship.
Good. What industry problem is the closed system group trying to address?
That's a great question. So actually technologies exist today wherein one can completely isolate almost all bioprocesses from the manufacturing environment that house these unit operations, and thus allow the industry to be able to manufacture in a controlled non-classified space, also called as CNC, without posing, importantly, risk to process and product quality and uphold patient safety. So some of the technologies are specifically closed systems, single-use, disposables, and also these are supported by the health agency guidances which espouse risk-based approaches for facility design. So recognizing the closed system benefits, what we continue to see within the industry is that many manufacturers continue to implement these closed systems within classified clean rooms, and at times also have additional segregation, knowing fully well that closed systems are successful in mitigating contamination and eliminate or reduce the environmental impact without relying on the environmental measures. So it's ironic that we still have such a belt and suspenders approach. Reviewing the current industry situation is where the BPOG closed system members we're convinced that as an industry, we are shy from fully realizing the potential of quality risk management principles, which are also supported by agency guidances, and hence we undertook an empirical data-driven approach, basically to collect data and see if one can manufacture employing closed systems in CNC space, and thus hopefully catalyze the movement of the reduced bioburden processing operations to areas of lower classification, desirably CNC.
Why did you set out to conduct these experiments?
Yeah, so to just expand upon the previous question, actually, while operating in the CNC space, which is the desired goal from an industry perspective for reduced bioburden processes, often when you use hybrid closed systems, the integration of the single-use components to stainless steel via sanitary connections require temporary opening of the otherwise closed system, followed by decontamination of these exposed surfaces in order to restore the system back to a functionally closed state prior to product application. So in these situations, it's extremely important to ensure that one has achieved the right level of bioburden reduction with regards to the particular process in question before you continue with the rest of the processing. So the connection points typically in the single use and the stainless steel interface are primarily can be steamed and pretty popular and known within the industry. However, from a facility design concept, use of steam would necessitate actually very cost intensive and I would say rigid infrastructural support. So in light of moving towards when the industry is actually moving towards flexible modular design or flexible facilities, in other words facilities of the future, requiring steam can be seen as an impediment for full realization of this modular ballroom concept. Hence, reviewing the current situation, the BPOG closed system team members wanted to evaluate if there are alternate measures to steam, and if one could actually clean the connection points in the hybrid systems using industrial chemical cleaning agents, which are commonly found, and still achieve an appropriate and acceptable level of bioburden at these connection points, as opposed to the conventional method of steam. Hence, this is actually so that we can enable further manufacturing in the CNC space. So this is what was a trigger for us to see whether it is feasible and acceptable to use simple chemical cleaning agents to reduce the bioburden in an otherwise closed system that would have been temporarily opened while making sanitary connections and during operations in a CNC space.
Okay. So what were some of your results?
Yes, sure. Actually, I'm very excited to share that because that's the primary thunder of my talk that I actually gave in this excellent event. Our studies actually set out, as I mentioned, to evaluate whether cleaning agents could help reduce the bioburden from after connections have been made in a CNC environment to acceptable levels. And so we designed and executed experiments wherein we intentionally actually contaminated the sanitary connections like cap and links, which is very common in industry operations or biopharmaceutical operations, and applied selective cleaning agents to see if we could get an appreciable bioburden clearance in the system. So we chose test organisms like Bacillus subtilis spores to represent a worst case scenario, as well as vegetative cells of Micrococcus luteus. Again, as I mentioned, the primary intent was to simulate a worst case contamination scenario at the point of the connectors that one would typically encounter while operating in a non-aseptic bioprocessing environment. Based on our experimentation, we were able to demonstrate for both Bacillus subtilis and Micrococcus luteus, which are the test organisms, that we were able to get a significant log clearance for these microorganisms. Basically, we designed the experiments where we contaminated these connectors with excess of the challenge organism. Then after the connections were made, then we flushed the entire system with each of the cleaning agent that was chosen for the study, following which we sampled for the residual bioburden by sampling the process stream as well as the surface of the contaminated connectors, and calculated the total bioburden log reduction in terms of bioburden clearance. And as I mentioned, for Micrococcus luteus, while all the cleaning agents were extremely successful and gave greater than 3 log reduction, interestingly, we saw that just hot Wi-Fi and Wi-Fi, just water and caustic, gave us a very high log clearance of greater than 5.8, which can you imagine where you just have a bag of water and you just roll it into your production facility and you flush your system, and there you go, bingo, you have a significant bioburden clearance. So that's really, really commendable and impressive. And in terms of Bacillus subtilis, again, the results were following the biology of the organism, which it is a gram positive spore. It's more difficult to kill. We did see between 2 to 3 log reduction for most of the cleaning agents. However, we got the most impressive results for hot caustic, that is caustic maintained at 40 degrees C, and CIP 100 and CIP 150. Again, these are very common cleaning agents that are prevalently used in the industry. We also saw additionally that when we compared the results for Bacillus subtilis contaminated on cap and links, that the bioburden clearance was significantly impacted by the design and geometry of the sanitary connections. So the overall takeaway from those was that one has to keep in mind that the bioburden clearance can be impacted by the design and the geometry of your connectors, the nature of the microorganism, and also the mode of action of the chemical agents. So to summarize, overall, we did meet the objectives of our experiment. We were able to show that one can attain actually a significant bioburden clearance, which could, it's greater than 3 log depending on the organism. In fact, we actually met the sanitization standards, because for sanitization, you at least need greater than 3 log reduction. So it is incredible. And to put these results further into the context, we were able to conclude that while operating in using a functionally closed system, operating in a CNC environment, one can, and having at least a 2 log reduction has lesser potential for contamination to the process versus performing open connections in a grade C area.
So what next?
That's a great question. So while having ended the experiments, now the entire group is focused on writing the technical paper, which will be published on the Biopharm Operations Group's website, following which we are also authoring 2 publications which will be hopefully appearing in peer-reviewed journals. So stay tuned.
What other objectives is the closed system working group addressing?
So we are also actually currently focusing on other aspects, but primarily it is a risk-based approach for viral segregation, and also using a risk-based rational approach for defining and determining the appropriate level of gowning for various reduced bioburden operations.
Exciting.
It is a very exciting time.
Aside from speaking, to conclude our discussion here, what brings you to the BPI conference?
Sure. So I've heard a lot about BPI, and I've been attending BPI for the past 3 years. And BPI, I recognize, is one of the largest bioprocessing event, and it is an excellent platform for all the industry folks, including myself, to learn what the current trends are in the industry, identify what the burning issues are, learn again from each other as industry experts, and collectively work towards a better future. So, and of course, to enable faster access of quality medicines to the patients. It also is a nice forum for connecting and collaborating with the industry experts to identify what the challenges are and develop industry best practices, which could in turn shape the health agency guidances.
Well, I'll look forward to seeing the publications, and thank you so much once again for telling us about your work with BPOG and with closed systems.
Sure. I'd really appreciate and thank the entire BPI organizers for this excellent opportunity, and thanks once again.